Okay. Right, welcome to the last session of our seminar series of this week and now of our program. Right, and let me introduce our last but definitely not least speaker, Professor Hernan Demba from University of California, Irvine, and he's going to talk about applications of change point methods in Brine signal analysis.
Yeah, thank you, George. So I'd like to take this opportunity to, now that I have in this room experts in change points methods, some of the open problems that I encounter in brain signal analysis. And so there'll be more questions perhaps than answers here. So here is some scientific motivation. So a lot of these were already motivated by Ivor earlier. So here is a cartoon of the EEG scalp topography. So we may have some electrophysiologic data. So these could be your EEGs or local field potentials and so on. And then you have these channels, let's say on the scalp. And we're interested in looking at essentially what's happening in each of these channels. And also whether activity in one channel either excites or inhibits activity in another channel, and in what way. Okay. So there are other spatiotemporal data. You have the hemodynamic. You see, so these are the fMRIs. We have the neuronal data. So this actually is a direct recording of the neurons, whereas the hemodynamic, these are simply indirect measures of neuronal activity. Okay. So the data from these experiments typically consist of, you have an external stimulus. So we have different types of stimuli. So one of them might be visual. You might see a flashing checkerboard, or auditory, where you would hear clicks throughout the experiment and so on. So Ivor talked about the stress type of an external stimulus. And of course, we might have some other types of data, personality, traits, genetic, social, environmental factors might be there. So here we do not observe directly what's going on typically in a neuron, unless you're looking at the spike train data. But what we see are the brain signals. So these are indirect measures of neuronal activity. So you have functional, fMRI, EEG, MEG, PET, and so on. And you also have structural. So this is the diffusion tensor imaging. Okay? So the ones on top, these are the, here you have data that evolve over time within an experiment. But with anatomical, it's not very likely that the brain structure will change over a very small period of time. So if a person, let's say, has Alzheimer's, then you see anatomical changes, but these happen over extended periods of time. And they also have acute outcomes. So during the experiment, sometimes subjects have a dial, and they, depending on whether they're very happy or they're very sad, then they turn that dial. Or they're excited or not excited. So you have that. You also have the skin conductance, where a machine actually measures how sweaty your skin is when while you're waiting for the moment when you might or might not get shocked, electrocuted. So I was in that experiment a long time ago. I just wanted to be able to empathize with the subjects in that experiment. So I thought, okay, why not? And there's the motor response, where you're asked to respond by moving something left, right, up, down, so on. So here is the unfolding picture in this experiment. So a stimulus is presented. The neurons respond. So I may not observe this directly. Then this response gives rise to what we call the brain signals, the fMRI, EEG. So we record these. And these signals also typically also map onto behavior. Okay, so how you respond to the stimulus. Now, these different arrows here might actually be modified or moderated depending on a number of things. So there's genetic factors, trait, social environment. So how a stimulus affects the neuronal response may be modified or moderated by some of these things. Okay? So in our research, we're interested in what happens to the signal when a stimulus is presented. So that's a very vague question, what happens to the process, okay? When you introduce some impulse. So to make it a bit more precise, you might be looking at, is there a change in the mean? So here is the, so prior to a signal, prior to a stimulus being presented, here is the mean level. As a result of a stimulus, there might be a change, a shift in the mean. So now there might be a change in the variance. So prior to a stimulus, you have a little bit lower variability in the signal, and then now you have more variability as a result of the stimulus. And here is where now a lot of us are interested in, so Ivor pointed this out earlier, where perhaps prior to a stimulus being presented, you have these three different regions interacting with each other in this particular way. Again, interacting, what does that mean? Okay, how do we measure dependence? Okay? And then how that interaction actually changes as a result of the stimulus. So there's a change in the interaction. So we're looking at these changes. Now, so as I said, in a typical data, you have your multivariate time series or spatiotemporal data. You have your stimulus, you have behavior, clinical, perhaps genetic as well. Now, in these experiments, there's a hierarchy. So there are many groups in an experiment. So you have typically patient groups, and you have the healthy control. And sometimes, even within a patient group, there are also subdivisions. Okay? So if you look at the autism spectrum disorder, there's autistic kids, for example, who are highly functional, those that are not so functional. So within the patient group, there's still a little bit of that subdivision. Now, within each group are many subjects. See, so now there's a hierarchy, and within a subject are many replicated trials. Okay, so you are exposed to that same stimulus or different types of stimuli over the course of the experiment. Okay? So think about this setup. So at least from my perspective, a Bayesian approach looks natural for modeling these types of data, but then you're dealing with very, very massive data, so you must have to, you need to do some simplification in order to implement your Bayesian strategies here. Or you can also put this under some mixed effects framework where you want to model variation across trials within a subject, variation across subjects within a group, and so on. Okay? So just because a subject is in a control group does not mean that every one of them responds to a particular stimulus in the same way. So maybe there is a quote unquote, a signature response for a control group. However, every control person will, his or her response may differ slightly, or maybe even a lot. It depends on the variability from that quote unquote signature population response. Okay. So what are here I'd like to, because we're in a change point workshop, point out a little bit some of the change point problems in brain data analysis. So one of them is identifying change points in this electrophysiologic data. So EEGs, MEGs, LFPs, these are data with excellent temporal resolution. You actually have a lot of, you observe a lot of time points here. Okay? So for example, with LFPs, you may have up to 32,000 observations per second, so that's a bit an overkill. EEGs, up to 1,000 observations per second, but sometimes they still subsample that. Now, so this is for temporal data. We're looking at changes over time. There could also be spatial boundary curves and surfaces. Yes. Local field potentials. So these are also electrophysiological recordings that are directly on the neurons. It's very invasive. So we have rats where we really insert the electrodes inside. Right? So not very many humans volunteer to be in that study. Not a lot of college kids in the US will. Now we're also interested in looking at these spatial boundary curves. Okay, so Ivor showed nice pictures about identifying areas in the brain, voxels that are activated, quote unquote. And so typically what's done there is they fit a model to every voxel. They fit a generalized linear model to every voxel, and then they test for the coefficients that correspond to a particular stimulus, and then they see whether that coefficient is different from the coefficient corresponding to the control. Okay? So, but it's a point-wise inference. Okay? Activation, is this voxel activated? Yes or no. This one, yes or no, and so on. Now, we might be interested in actually looking at spatial boundaries. So you're looking at this group of voxels are activated. If I increase my voxel, do I see activation? If I increase further, do I stop? When do I stop? When do I stop increasing my starting volume? Okay? Kind of like in time series, this is stationary, and this is one process. If I move a little bit, 2, 3, 4 points, the process changes, and therefore I say that a change point has occurred. So here we're looking at the volume in space. Now, this is also temporal, but it really takes into account cross-dependence between regions or between channels. So here, this is what I'm working on currently with Christina and Gorostieta and Rainer von Sachs. So if time permits, I'll describe this a little bit. But these are the primary questions that are pervasive within the community, and I'd like to spend time on those. So here is an example of an electrophysiological data that we have. This is a visual motor experiment. So the PI here is Jerome Saint. So the experiment is this. So we have participants. So these are college kids who participate in the experiment. You know, we give them free pizza if they participate. So what they see here is that there's a screen, and on the screen they're given instruction: move the joystick to the right or to the left. Well, really there are more than just 2 directions. There are 6 or 8 directions, but I'm just focusing on right and left. Okay? And so as you can see, this is in a bigger grant on the goal of the grant is much more grand than what we could really do. So the whole point here is motor intention. From the time that the subject sees the instruction left or right to the time that the subject actually executes the instruction, there is that particular period where the brain processes that information and decides what to do left or right with the joystick. So that is a motor intention period that we were interested in, but it turned out to be very, very difficult. So we had to calibrate our expectations and redefine our new aim. So in the end, we are able to produce something. Okay? But so that's that's the grander goal. But here for now, we just want to look at what's going on in the brain via looking at these EEGs in this particular visual motor task. So they see the instruction, visual, and then they execute motor. Okay? So here, there are 64 channels total, over 100 replicates for the right direction and over 100 for the left direction. And one trial is 100 millisecond worth of recording. Now, some questions here are, is there a pre versus post-stimulus difference in a brain response? So if you look at stimulus at time 0, you look at EEGs the EEG recording prior to that and after that. Is there a difference? So is there a difference in the post versus pre-stimulus? Okay? So when we think about is there a difference in the brain response, again, we're looking at, are there changes in the mean level? Are also within and between channel dependence. So for quite a long time, we're looking at only coherence or cross-coherence, correlation or cross-correlation as our measures of dependence. Okay? But these are typically just linear measures of dependence. And for us to say that I'm able to capture everything in brain signals by looking at linear dependence is a bit of an oversimplification. Okay? And in fact, when we were doing some classification studies, really we were pointing towards higher order types of dependence that can best discriminate between signals. Okay? So anyway, so looking at the point here is that there is the point in time when a stimulus was presented. Is there a difference in pre versus post? However, actually you might not see the changes right at when the stimulus was presented, because it takes some time for the brain to process that information. So there is that change point problem. Okay, up to what point in time post-stimulus presentation might actually we see the change? Now, one also would like to look at, is there a dynamics or an evolution to the process post-stimulus period? So it's possible that there could be different, I would say, regimes going on post-stimulus presentation. It's not only one change. Maybe post-stimulus presentation, there could be many changes as well. Okay? So the process evolves. There is dynamics in the process post-stimulus presentation. Okay? And then here is another very important concept that people are looking at, is the consistency or lack of it, variability in brain responses across replicated trials. So I think you saw this in Ivor's work earlier, where there were 23 subjects, and then for the first change point, there appears to be a lot of consistency, but you didn't see that. You didn't see that towards the end. Okay, so there is still that variability across trials as to where the change point might be. Now, the variability could be, of course, due to a lot of things, right? Number one is, there's the brain is not an IID kind of a machine, right? So every now and then it changes. Or it could also be due to just really measurement error. So I would think that the precise timing of the stimulus presentations, I must say they're not quite as accurate as German trains. So, there's going to be a little bit of shifting here and there. Okay? So it's not perfect. And so the variability might just be due to that, but I think largely due to the fact that the brain is not a static object. It's allowed to, it has a variable responses even to the same stimulus. So I want to look at the distribution of change points, and also you want to look at the variability in the change points for different conditions. Okay? Condition A, are my change points more or less uniform? Are they more or less consistent? Condition B, is it all over the place? So in my collaborative work with Claudia, in fact, we saw a bit of that variability in the change points according to different conditions. Okay? So by the way, that's the paper with Claudia. So that discusses some of these findings. Now here's another experiment, and this is on looking at the neural correlates of decision making. So decision making, it's a bit hot now within neuroscience. So we're looking at is there a neuronal basis for decision making? So this is with my collaborator David Moorman from UMass Amherst. So the goals here are to model how the neural systems assimilate information in order to produce a decision. So in this experiment, we have rats, and the rats are trained so that when they see two levers inside a cage, then they would press on a lever, and then they would get a reward. Okay? So this is a common paradigm in animal studies. So monkeys, they also expose monkeys to this. Now, so the rats see two levers, and one of these levers, they know that when they press the lever with very high probability, they will get a reward, but that reward they know is going to be small. Okay, so now another lever with a lower probability that they will get a reward, but when they get a reward, they get a jackpot. So now a rat has to decide, for this moment, am I going to go for the sure thing, or am I going to be adventurous and maybe get $1 million? So I don't remember exactly what the reward is, but for our monkey study, the rewards actually were a fruit juice or a beverage. So the monkey is trained, and they know exactly if the monkey's favorite beverage is a root beer, then that's going to be the reward. So the monkey's favorite is apple juice, then that's what the monkey gets. And the So the rats and the monkeys, they're humanely, I shouldn't say starved, but they're humanely put in a situation where they will have an incentive to go for the reward. If they're hungry, if they're full, they're not going to care. But if they're a little bit thirsty, then they would want to, or hungry, they would want to care to make the best choice for them. So here, you want to choose between a low risk, low reward choice versus a high risk, high reward choice. Now, for a long time, neuroscientists only look at single neuronal responses. That's what they've been used to. But now you really want to look at the interactions between these neurons, particularly in the medial frontal cortex of the rat's brain. So now our data here consists of spike train and local field potential. So there's a bit of a pre-processing that goes on here that does not involve statisticians. So we have, I think at some point we have to be more involved in what really happens to our data to the point that I get it. I need to know what has gone. But my understanding for now is this. So you have the analog signal, they're filtered, digitized, and so there's a high-pass filter, and when the high-pass filtered signal exceeds a particular threshold, then they declare a spike at that time bin. So it's a 1-0 thing. That's a spike train data. Okay. And then we have lower frequency signals. So lower frequency is very relative here because lower frequency for LFPs means 0 to 300 hertz. So that's still a lot compared to fMRI, where you only observe 1 to 3 hertz probably. Okay. So here, then you have the local field potentials. So this is in a way, a multimodal data because now you have your binary data, time series of ones and zeros, and here you have your continuous valued time series. So again, here is a picture of the micro wire array implanted in the brain of a rat. So David Moorman actually he does the surgery, surgical incisions himself. Okay. So here is a picture of what we would like to do. So for example, here is neuron 1. There's a sequence of ones and zeros, spike or no spike, and also in an area very close to. And also we have an LFP recording that's believed to be mostly associated with neuron 1. There's still a little bit of smearing when it comes to LFPs because it's a field, and therefore whatever activity that I record there is most likely due to the neurons closest to it, but there could be effects from other neighboring neurons. Okay? So there's a bit of that. That's why it's a field. But we will tie this activity with neuron 1. So you have this continuous valued time series. There's neuron 1, there's neuron 2, there's neuron 3. So in the past, all people did was simply analyze just what's going on in neuron 1, analyze a spike train here, spike train here, and spike train here. And then very often they would actually, what they do is they would only look at the spike trains and then draw a histogram trying to understand where the spikes occur and the density of these spikes. Okay. So people have done Poisson processes for modeling, modeling these neuronal spike train data. But now what we'd like to do is look at whether past spiking activity in neuron 2, if that actually excites or inhibits spiking in neuron 1. Looking at these between neuron dependence, and whether past oscillations in neuron 2, so you have your alpha bands, gamma bands, and so on, whether an increase in alpha band or an increase in gamma band in neuron 2, how does that influence the probability of spiking in neuron 1 in the future? Okay, so we're looking at binary time series and multivariate time series. Okay? So this seems to be quite natural for us to be thinking about, but this has not yet been done within the neuroscience community, although I think there is one group now that's also starting to do this in Germany. It's still in a very early stages. So here we're looking at the activity of an ensemble of neurons, and they, of course, will provide more information than only that from a single neuron, and therefore there's a motivation to look at this ensemble population instead of just single neurons. And as I was saying earlier, these are our questions: how spiking activity in one neuron might be influenced by spiking activity in other neurons, and also looking at the spectral powers in other neurons. Now, there is an evolution of course, brain responses across trials. Okay, so here is trial 1. We have 1 or 10 seconds worth of recording for spike train data and also LFP data. Now, how do we actually model the variation in responses across trials? So I believe that Mark Feckas talked a little bit about this earlier in the week. Maybe I'll just recap. So if you have the multivariate time series data, Y, which I collect at trial R, at time T, I can model my YR, so this is the data at trial R, equals some mean function that's specific to trial R, plus some random term that's also specific to trial R. If I want to model variation in this mean function responses, then I can decompose f of r into your global mean, and then you have your trial-specific deviation from that global mean. So this is well known in functional mixed effects already. Okay? And so now, however, in classical mixed effects models, you have many subjects, right? And you're looking at the variation in the responses across these many subjects. And typically, for subjects, there's no temporal ordering. Subject 1, subject 2, and so on. I think of these as just independent. But here, the trials have a particular temporal ordering, so that the functional responses between trials that are close to each other, the functional responses of trials that are close to each other are expected to be a little bit more similar than those that are from further apart trials. Okay? So there could be perhaps some AR structure in these parameters. Okay? So again, there are many ways to, of course, characterize this. You can put this you can model your f of t in terms of some, spline functions or wavelet functions, and so now you have this f, and then you also have this. You can also model this in terms of splines or wavelets, but then you allow your coefficients to vary, right? To be random, vary across trials. But then how they vary across trials, you want to put a little bit of a temporal structure in that. Okay? So that's this is an open problem. Okay? So now to the next problem of change points, but here I would say boundary surfaces. So we're saying that, okay, in this part of the brain, we have activation, this part, no. Although I must say that in a lot of these cases, you might see a smooth transition between highly activated, mildly, and then none. Okay? So it's not a very abrupt change point. I think that's true for most brain responses too, and it's for that reason why in fMRI, we don't model here is a stimulus, and then no stimulus, stimulus, no stimulus. We convolve this with the hemodynamic response function so that the bold response is actually smoother. It's not a yes-no thing, so it's slowly going up to a 1, it decays, slowly going up, and so on. We convolve this stimulus indicator with the hemodynamic response function. So now here, we're dealing with fMRI data. It's massive spatiotemporal, really more massive in the spatial dimension. There could be 100,000 voxels, and number of time points might be in the hundreds. Number of subjects, I would say it used to be that if you have 10, that's a lot, but now we're going up to hundreds. So ADNI data has probably 300 or even 700. So we're having more and more of these. The problem, though, is that there is a lot of variation between magnets. Okay? So you see, there is a traveling phantom experiment where they're not phantom necessarily humans, but they call it a traveling phantom. So there's 5 or so subjects. They travel within the Northeast, and then they have their fMRI scans at many different locations. And there is a lot of variability even if they use the same scanner. And that's the same geographical location as well. Okay? So the question is, how do you actually do a meta-analysis of all of these data collected from different places? Okay? I don't think that's been solved. But it's something we need to do because it's hard to recruit many subjects only in one site. You have all these multi-site studies. So it's not a change point problem, but nevertheless, that is a very important problem that I don't think has been solved. Now, so here, when we're doing, okay, so perhaps I already mentioned this earlier, that this is an indirect measure of neural activity. So as I was saying, it's great spatial resolution. So you have even 1 millimeter cube in resolution. I ever mentioned about 3 times 3 times 3 a millimeter cube, but now you can go up to 1 millimeter. You can make it as of course, the price to pay is that if you have more spatial resolution, then it'll take longer to scan and so on. Okay? And very often too, I think you mentioned this yesterday, where you have data from individual nucleotides, and then in the end you just average. So here we don't do really time series on an individual voxels when we do connectivity. We look at regions of interest. So it's kind of like a waste in the effort to create data with very high spatial resolution. But in the end, you look at a coarser level anyway. Okay? Anyway, so that's just an aside. So you have a temporal resolution of 1 to 3 seconds. So the aims in fMRI studies are activation, identify which regions respond to a stimulus, and also connectivity. So very, very vaguely here, how do brain regions communicate? So when people look at connectivity, of course, they look at cross correlations, light cross correlations. We also look at coherence at very low frequencies. And the word connectivity is very fluid. So when you analyze your data, you have to be very specific as to what your metric of dependence is, what it is that you're using. So for modeling activation, I mentioned this a little bit earlier, is that the standard approach is at every voxel, you fit a generalized linear model, okay? And then you perform your inference. And very often this ignores spatial correlation. Even though the data you get has undergone a lot of spatial smoothing already to begin with. This part is ignored. Now, but that's the standard approach. And also I've seen work that's done on the whole brain independent components analysis, but then typically that also does not use spatial information. So if I have time series and 100,000 voxels, they really don't care about how those voxels are arranged in space. I can permute them and get the same ICA results. Okay? So they do not take the spatial information into account. But these are the standard approaches. I believe that we're still at the very beginning of the very beginning stage of really how defining what is the optimal way of analyzing the data. So there's still a lot of work to be done. More current approaches here is that we use regions of interest instead of voxels. Okay? So when we do this, of course, we have a reduced spatial resolution. Instead of having one, then you have a bigger volume. But then that's a way to make the computation a bit more tractable. Okay? It's hard to really invert a 100,000 by 100,000 covariance matrix. So we have to find ways to deal with the problem. Now, here is a problem. The ROIs are predefined typically. Okay? The boundaries are predefined. So in the end, the conclusion is, so here is what happens typically in this collaboration. And so your neuroscientist identifies voxels that they believe are highly activated. And then from those voxels, they form a sphere. And from that sphere, they extract the time series, and then they average the time series, and then that becomes the quote-unquote data for that region. Okay? Now, sometimes the regions are predefined anatomically. So prior to any experiment, so there is the what they call the anatomic parcellation of the brain into 107 areas. Okay, so and then for each of those areas, they obtain the average time series. Right? But the point is that these are predefined boundaries, and what we like is to actually obtain those boundaries using the data. Okay? So we're looking at a point where how do I segregate here is my collection of contiguous voxels that are activated. I want to be able to expand that using some spatial change points approaches. So that I have not seen done yet, but I sort of vaguely remember John. That's why I was asking John and Claudia if they have done work on this, because somebody mentioned this, but Claudia said no, so maybe it was John, maybe it was somebody else. But people are thinking about it, but it's not been done. But I think it's interesting. Not let the parcellation dictate to me how I define my boundaries. I want to be able to use my data to define my boundaries. But I don't like to do it in a point-wise manner where, yes, activated, no, activated, no. I want to be able to do a more smarter thing than that. So as I was saying earlier, maybe one can start from a small 3D region and then keep on expanding until you should not expand further, because at that point, the process is already different. You've changed a boundary. Of course, to do this, how do I say that the process is different? Therefore, I need to have some metric, and once I have a metric that's a measure of this similarity, then I need to have some threshold as to what is a significantly different process, and so on. So. And so that's one thing. The other thing is we're dealing with very massive spatial data here. Okay? So we'd like to be as sophisticated as we can, but we have to be also smart and really take this into account. Maybe to the extent possible, use efficient algorithms, and also use some prior information as to how you would actually do the analysis. So in my recent experience with one of the prominent persons doing imaging genetics for psychiatry, so what he wanted me to look at was connectivity between brain regions, comparing the schizophrenics with the healthy controls, and now looking at how genetics might play into that. And so we have some, mixed effects vector autoregressive model. We can, in theory, incorporate some, genetic data, broadly speaking. But I wanted to do it on predefined regions of interest. I don't want to use the entire brain. Okay? And he didn't want to do that. He said, why don't you want to use the entire brain? And I said, well, if you want me to use the entire brain, then I will be using some penalized regression methods where I ignore all the information that's available out there and then really let this criterion tell me what the output is. Question? Okay. And I said, so perhaps what we should do is really, you're looking at schizophrenics, you must have over 30 years, have some idea about what regions are actually interesting here. Why do I want to look at the occipital? Why do I want to look at the parietal and so on if you think they're not going to be implicated for these diseases, for those particular processes? And so my sense is that even though they've been in the science for a long, long time, at this point they're at the very edge of what they know about their field. So I used to think that they have all these built-in theories, but these theories that they formulate are actually based on an accumulation of little empirical evidences, and then they build a theory out of that. But at this point, there's not a coherent set of empirical evidence, and therefore they cannot have a physiological theory for this. So that's why he is so willing to go into this very data mining type of an approach versus, let's focus on these regions. Anyways, this is my experience. I'm sure others have their own take on that. Now, for activation, how do we combine results from many subjects? So, perhaps the boundaries might differ as you go from one subject to the next. Okay? So we need to have some notion of perhaps a distribution of boundary surfaces. Okay? So here's some sort of average boundary, but then there's a bit of a spreading out. There's a lot of variability across subjects, and maybe for one patient group, that distribution might be very tight, but for another group, it may be a bit more dispersed. Okay? So some notion of a distribution around some center boundary, okay? Some central boundary surface. So modeling connectivity. So here, one of the issues is scientists want to be able to make statements about physiological causality. But when we're fitting vector autoregressive models and so on, all we can say is that it's just Granger causality, right? Where knowledge of one variable helps me to predict another in the future. It's just a Granger causality. It's not physiological. I cannot make that statement. Although they'll try to actually twist your arm and saying, can we actually say this? Is that your experience, Ivor? They want to be able to make that strong conclusion, but no. So really, with Granger causality, all we're saying is that knowing a bold response in one region helps me to predict the bold response in another. Okay? Helping me to predict does not mean that activity here is causing the activity in the other. Okay? Those are two different things. And so, in fact, in fMRI, a temporal ordering in the bold response or in the neuronal response might not really match the ordering in the neuronal response. I've seen a lot of simulations where in the neuronal level, one precedes the other, but because of the timing and the convolutions in the fMRI, the ordering can actually be reversed. Okay, so now, what are the metrics for modeling connectivity? So we have the cross correlation, we have the cross coherence, typically only at low frequencies, and then we have parametric VARs. So these are the typical measures of extracting typical metrics for connectivity. Okay? Now, connectivity is of course dynamic in time. Here is where we want to look at changes over time with respect to connectivity. So I will show this where you have these graphs of 15 ROIs, and depending on which time point you're in, you get certain connections between nodes, and then you lose them, you gain them over time. Okay? So it's changing over time. We need to identify shifts in connectivity, changes following a stimulus. So we have some work on this on the mixed effects vector autoregressive. Okay? But moreover, how connectivity can also change across space. So let me, perhaps you might look at identifying the boundaries. If you have two disjoint regions, you want to identify voxels that are highly codependent with another set of voxels in another region, and then do some boundary surfaces based on that, instead of just looking at the boundary with respect to the mean. Okay? And then accounting for variation between subjects, very tricky thing. So for example, you might think about here is a population graphical model where A and B are correlated and so on. But then how do you actually model the variation of these graphs across subjects or across trials? Okay? So it appears that perhaps a Bayesian approach might do it, but it's still open. So there's a lot of work going on, but it's still very much open. So we have about 15 more minutes. I don't know if I should continue with this, or maybe it's a good time to pause for a little bit, and if there are any questions, I'd be happy to on what I have talked about so far. Yes, please.
With regard to the question of of regions rather than individual voxels, how does Worsley's work fit into this question?
So Worsley's work is still very much voxel based. So what he did was he obtained a threshold treating, so you have a map of T statistic or F statistic values, and then treating that as a realization of some random field, initially Gaussian, and then with Jonathan Taylor they expanded that a little bit. And so then looking at the threshold, applying a threshold on that to identify these specific regions. I'm not sure whether the contiguity. Are you familiar with this, John? The technical details of the contiguity in the Worsley work.
Yeah, I mean, I think possibly more sort of related to the region idea is that he's had some ideas about scale space. So you actually sort of looked at things on a very voxel level and then you expanded them out. And in some sense, you tried to find which level of scale is the most representative of your data. And in some sense, that could be seen as being very much related to trying to define regions. I mean, I don't know that there was any sort of necessarily anatomical information built into how to define those scales. And I think there's quite a few people who've looked at, or recently looked at, sort of ways of trying to define regions in space, but they usually take a more computational approach of picking some kind of way of expanding or contracting space, and then using that to define what the regions are, rather than any kind of principled anatomical.
So perhaps in that multi-resolution type of an approach, although in principle I like that, but I still like the idea of extending what we know in temporal data, where as I expand my little block, do I see a change?
Another question with regard to the kind of signal you look for. There was a period of time where diffusion tensor imaging was very fashionable.
Yes.
Is that still?
It still is. It still is. And so Ivor talked a little bit about that, looking at really fiber connectivity. So DTI itself is as a tool is interesting. The only problem is that people look at voxel-specific eigenvalues, and then they look at measures of what they call the fractional anisotropy, where they look at the eigenvalues, and then what they do is they look at the average of these eigenvalues or the variability of these eigenvalues, and then based on that, they give you an idea about the potential path of a water molecule from one region to the next. But that is still very much into play, and in fact, that will be a good anatomical information to build onto your functional. Okay? So in the presence of these structures, then the probability that, the likelihood that two regions are actually going to be functionally connected is also high, given that they have the structure present.
But that's going to have other scale issues, right? Putting together fMRI and DTI and registering and being
Exactly.
spatial scales.
Exactly, yes.
Especially as the brain is quite spatially homogeneous.
Exactly, yes. So what I've seen done so far is a very crude way of doing that, where suppose I have predefined ROIs now from fMRI, then I have my time series for each of those ROIs, and then I look at my DTI data, and then I look at I define some path between my ROI1 and ROI2, and then using DTI, and then take some sort of a metric on how the strength of that anatomical connectivity, and then use that as a prior for your functional. But it's very crude.
Estimating that path is still not very well done.
Exactly.
I mean, they like to seed things and see where the particles go and blah, blah, blah.
Yeah.
But they don't have a good notion of how their uncertainty in the inference of their directions reflected when they do diffusions. And usually you get different answers if you start from one or the other.
Exactly.
And that has no anatomical interpretation. Right?
Exactly. Definitely. Yes. Yes. Yep. Yes.
When you said you wanted a distribution on surfaces or shapes, you want to have something a bit like a normal distribution, so you want to say this is a mean shape and then this is a
Yeah, to be honest with you, I just have some idea in picture, but I don't quite know how to make it really rigorous. So what I had in mind was something like this, where if I if I suppose this is my curvature, right? This is my boundary surface, and so I want to be able to look at some spread around that. Just an idea about, so here is one possibility, but maybe for, you have a bit more of uncertainty here versus here. I'm still not sure how to make it rigorous.
So could you take your mean shape and then can it be discrete? So can it be a set of points rather than...
I think you can, because all of these are discretized voxels anyway. Yeah.
Okay, so if you took your mean shape and then if you do the discrete Beltrami on that, take the eigenfunctions, and that gives you a basis for the shape.
Yeah, I think that's possible.
Could you just put a distribution and then expand out your mean shape in that basis?
Yeah.
And then put a distribution on what you think the coefficients around that are, and that might give you a...
I think that's possible. Yep. Of course, you want to be able to allow for the spread might also change across a surface, right? The variability might change across the surface.
Yeah, and it won't, because it's not localized.
Yeah. But if you can have something like that, I think that would be nice. That would be a big step towards, starting with, I know my regions, and I want to be able to define those. Okay. So maybe just a very quick thing on this, because this has been what's on my mind. So we talked about correlation, we talked about these as measures of dependence, but now looking at coherence is a measure of dependence because you're looking at interactions between oscillations at different channels. Okay? So I think with only 3 minutes, I'm going to just post a lot of pictures here. Okay? So here is my time series 1, time series 2. I can decompose this into low frequency, high frequency waves, and so on. And cross-coherence, in a classical sense, is looking at the squared cross-correlation between these components. So you apply a filter, you cross-correlate, you square them. That's the coherence between channel 1 and 2 at frequency alpha. Okay? Now, so that's classical. However, there is now an interest in looking at how does the alpha activity in one channel, how is that codependent with gamma activity in another channel? There's an increase in activation here. Is that contemporaneous with an increase or a decreased activation in a different frequency band in another channel? So this is now the dual frequency coherence. Okay? So in fact, now looking at another extension to this is that how the alpha and the gamma relate to each other might actually also evolve over the course of a trial. Okay? So looking at, we have the stationary processes, Cramér representation. Priestley came, look at the how the spectrum might evolve over time. Dalhouse worked on the asymptotics, built a framework for which we can show consistency, and we'd like to be able to parallel that. Okay? So how this process evolves over the course of a trial, looking at how alpha and gamma are connected with each other. Okay? So here again is a picture where under stationarity, the frequencies are actually uncorrelated. So you have a diagonal, you have a diagonal covariance matrix in a way. And under stationarity, at any time point, this diagonal matrix is not changing. The covariance and the frequencies is the same. It's diagonal, it's not changing. For locally stationarity, then locally it's diagonal, but over time it could change. Okay? The decomposition of signals over time may change. That's in the locally stationary world. Now for harmonizable processes, then you have omega 1 and omega 2. You have dependence over all frequencies, but at any given point in time, that dependence between omega 1 does not change. Okay? So paralleling the Dalhouse and Priestley innovation here, we're looking at a locally harmonizable process where as you go from one time point to the next, it's a two-dimensional covariance matrix. It's not diagonal, but it could change over time. So that's just really the nutshell of what we would like to do. So I think I'll just end here. So as you can see, I've been very fortunate. I really am been very fortunate to have had the opportunity to exchange ideas with a lot of people that I enjoy being with, both on a professional and on a personal level. So I've perhaps because of a lot of these collaborations, I have had my biggest share of a lot of wines. Bottles open, a lot of beers. So you can imagine it's multiplied by 10, right? So, and of course, a large number of my collaborators are actually here in this audience. And so thank you to all of you. And finally, I think all of us feel the same way that we'd like to really congratulate and thank our organizers, John, Idris, Paul, and George, for the wonderful workshop.
Suggestions, because this is the last talk of our program.
Well, I have one more question to answer, which was about the coherence, cross-coherence. You mentioned you mainly looked at low-frequency cross-coherence. I was wondering what the reason for that might be. Is it a sampling issue, or is it
for fMRI? It is both a sampling. It is a sampling issue. And also the fact that we're dealing with a kind of data where you don't see a lot of variability across zero with fMRI. So that's not true for EEGs, but for fMRI, it's more of a low frequency signal. Yep.
Any other collaborators? Yeah, Dr. London, right? So if not, please join me to thank Fernando and all other speakers of the week and their own programs for their talks.